Hi, today I'll be presenting our protein synthesis model. Protein synthesis is the process cells use to make proteins, and it happens in two main steps: transcription and translation. Transcription happens inside the nucleus of the cell. In our model, the yellow piece represents RNA polymerase. RNA polymerase moves along the DNA and builds a strand of mRNA using complementary base pairing. That means guanine pairs with cytosine, cytosine pairs with guanine, adenine pairs with uracil and RNA, and thymine pairs with adenine. So, you can see the colored beads in our model represent these nitrogen bases. Once the mRNA is made, it leaves the nucleus, and moves into the cytoplasm. Next is translation. Translation happens at the ribosome. In our model, the ribosome is shown surrounding the mRNA strand. The ribosome reads the mRNA in groups of 3 bases called codons. Each codon matches with a tRNA molecule carrying a specific amino acid. The tRNA has an anticodon that pairs with the codon on the mRNA. As the ribosome moves along the mRNA, amino acids are joined together to form a polypeptide chain, which will eventually fold into a protein. On the right side of our model, so left of the camera, we show a comparison between a normal cell and a sickle cell. The top pipe cleaner represents the normal polypeptide chain. The bottom one represents the sickle cell polypeptide chain. In sickle cell disease, a mutation changes just one base in the DNA sequence. This changes one codon in the mRNA, which changes one amino acid to be different in the final protein. Even though it's only one change, it affects the shape of the hemoglobin protein, which causes red blood cells to become sickle-shaped. This shows how one small mutation in DNA can lead to a major change in protein and affect the entire organism. Overall, our model demonstrates how DNA is transcribed into mRNA, how mRNA is translated into a protein, and how a single mutation can change the final protein product. Thank you for listening.